Hello everyone, welcome to ITLS Academy. I am your mentor Arnab and today's topic of discussion is about the DNA replication in prokaryotes and eukaryotes. So at first we will see an animation. Now we all know that the double-stranded DNA is need to be unwounded and the work of unwinding is done by the enzyme helicase. So here it is the enzyme helicase and it is binding at a particular site, that is the origin of replication. So helicase is binded here. Now see that helicase is unwinding, that means it is breaking the hydrogen bonds, and here the single stranded binding proteins are binding to the unwounded strands, which is ensuring that those strands doesn't form the double helix again. It's clear now? Okay, we will moving to the next slide. See the difference in prokaryote and eukaryotes. Okay, see here in case of prokaryote there are multi proteins present, that is DNA A, DNA B, DNA C. Okay, so at first DNA A starts the replication's initiation. It binds at the 9' sequence. After that it binds to the 13' sequence and start melting, and it is ATP dependent process. Okay, after that DNA B is the helicase enzyme. So the enzyme DNA B acts as a helicase enzyme and it is binding at the site where the double-stranded DNA will start unwinding, and another protein is required to load the helicase into the replication site, and that protein is DNA C. Okay, now next is SSB protein. So after helicase has started the unwinding, the SSB proteins are binding to the unwinded DNA. In case of prokaryote, the DNA G protein work as a primase and it synthesizes a RNA primer for initiation of DNA replication. Okay, now here we will learn about the same process which is done on the eukaryotes. In case of eukaryotes, MCM works as a helicase, RPA or replication protein A work as a SSBP, means single stranded binding protein, and DNA polymerase alpha work as a primase. So here in case of eukaryote, the RNA primer is synthesized by DNA polymerase alpha, and in case of prokaryote, the RNA primer was synthesized by DNA G protein. Okay, it's clear the differences between the initiation of prokaryotes and eukaryotes. Okay, now we will be moving to the next slide. Now as we have learned earlier that at the time of unwinding there is a pressure created on the DNA double helix, and that can cause supercoiling, and that supercoiling is removed by the enzyme topoisomerase. There are two types of topoisomerase enzyme found. One is type 1 and one is type 2. Generally type 1 topoisomerase enzymes causes a single strand break. Then it passes those strands and the pressure is released here. Okay. Now in case of type 2 enzyme, they causes a double stranded break here and the pressure is released. Okay. Done. Now in case of type of 2 enzyme, there are two subunit. The type 2 topoisomerase is also known as DNA Gyrase, and there is two subunit. One is Gyre A and one is Gyre B. In case of Gyre A, it done the mechanism of cut and paste, and Gyre B is involved in the ATP hydrolysis. Fine. Now there are few antibiotics which targets the DNA Gyrase enzyme, that is the norfloxacin, ciprofloxacin, or the we can say that quinolone antibiotics or novobiocin. Those antibiotics generally targets DNA Gyrase and inhibits their activity. Fine. Now we are moving to the next slide. Now in this slide we will learn about the elongation process of DNA replication. Okay. Now the upper strand is the leading strand and the lower strand is the lagging strand. In the leading strand we can see that the replication is continuously happening. Okay, let me enable the pen. So it is the leading strand. Here the replication is continuously happening. In the lagging strand, it's an interesting matter. Just see here, the RNA primer is synthesizing by the enzyme primase, then DNA polymerase 3 is replicating the strand. After that, the RNA primer is being removed by the enzyme DNA polymerase 1 and the gap is sealed by the DNA ligase. So in the lagging strand, that is an interesting matter. At first the RNA primer is synthesized by the enzyme primase, then DNA polymerase 3 is synthesizing a small fragment of DNA, then RNA primer is removed by DNA polymerase 1 and those fragments are connected together or joined together by the enzyme DNA ligase. Fine. Those fragments in the lagging strand is known as the Okazaki fragment. Okay. Now in the lagging strand you can see that the replication is discontinuously happening, and in the leading strand you can see the replication is continuously happening. Okay, for that reason DNA replication is known as semi-discontinuous process. Now in this slide we will compare the elongation process in prokaryotes and eukaryotes. So in the prokaryotes we have seen that the DNA polymerase 3 is catalyzing both the leading strand and the lagging strand, and the RNA primer gets removed by the enzyme DNA polymerase 1. Okay, now in eukaryotes it's bit of complicated matter. Okay, there are several polymerase which controls the replication. Here the DNA polymerase delta and DNA polymerase epsilon catalyzes the replication in lagging strand and leading strand respectively. That means DNA polymerase delta catalyzes in the lagging strand and DNA polymerase epsilon catalyzes in the leading strand. And we have discussed earlier that in case of eukaryotes the primase activity is shown by the DNA polymerase alpha. So at first the DNA polymerase alpha is synthesizing the RNA primer, and after that it is switched to different other polymerase, that is the DNA polymerase delta and DNA polymerase epsilon. This is the process. This process is known as polymerase switching. Now here comes another fact, the removal of RNA primer in case of eukaryotes. In case of prokaryotes there was DNA polymerase 1 which was removing the RNA primer, but in case of eukaryote it is a bit complicated matter. At first the RNase H, that enzyme, removes the RNA primer, but it has the capability to remove only interaction between two RNA molecules or the two RNTPs, ribonucleotides. It cannot keep the hybrid of DNTPs and RNTPs, that means the deoxynucleotides and ribonucleotides. Okay, so the last nucleotide, that is the RNA DNA hybrid, and that nucleotide is removed by the enzyme fen endonuclease. Okay, so it is clear that the difference between the elongation of prokaryotes and eukaryotes. So now we will learn about the termination of DNA replication. So here you can see that the particular protein, that is the TUS protein, is binding at a termination site that is known as TER termination site or TAR termination site. Whenever helicase enzyme reaches that region, it cannot pass through here. So the unwinding process is not done. Due to that reason, DNA polymerase cannot synthesize the new DNA again, so helicase cannot unwind the DNA again, and replication is stopped here. Okay, so we will practice some questions regarding the DNA replication today, because it is the end video of the DNA replication topic. So we will practice some questions here now. Now see here at the first question, it is saying which of the following is true about DNA polymerase. First point is it can synthesize DNA in 5' to 3' direction. Yeah, that is a correct statement. Second point is that it can synthesize DNA in the 3' to 5' direction. No, it is the wrong statement, because DNA polymerase always synthesizes DNA in 5' to 3' direction. Third option is that it can synthesize mRNA in 3' to 5' direction. No, DNA polymerase synthesizes DNA, not RNA. Fourth is it can synthesize mRNA in 5' to 3' direction. No, that's also wrong. So the correct answer is option A. Now in the second question, it is asking that which of the following reaction is required for proofreading during DNA replication by DNA polymerase 3. Now we all know that DNA polymerase 3 has two properties. One is 5' to 3' polymerase activity, and another one is 3' to 5' exonuclease activity. Now you require exonuclease activity for the proofreading, so the proofreading is done by the 3' to 5' exonuclease activity. Now in the third question, the reaction in DNA replication is catalyzed by DNA ligase is. First option is addition of nucleotides to the leading strand. No, this is done by DNA polymerase. Addition of new nucleotide to the lagging strand. No, this is also done by DNA polymerase. Third one is formation of phosphodiester bond between the 3' OH of one Okazaki fragment and the 5' phosphate of the next lagging strand. Yes, this option is correct. Ligase joined those Okazaki fragments. Okay. Option D is base pairing of template and newly formed DNA strand. No, the base pairing is not done by DNA ligase. So correct answer is option 3. Now look at this question. Which of the following enzyme remove supercoiling in replicating DNA ahead of replication fork? First one is DNA polymerase. No, DNA polymerase doesn't do that. That enzyme add DNTPs to the new daughter strand. Second one is DNA helicase. No, that enzyme is involved in unwinding of DNA. Third one is primase. No, that enzyme catalyzes the RNA primer which is required for the DNA replication by DNA polymerase to add DNTPs to the daughter strand. Fourth one is topoisomerase. Yeah, this is the correct answer. Now, which of the following enzyme is principal replication enzyme in E. coli? First one is DNA polymerase 1. No, it is not because it has a low processivity. Second is DNA polymerase 2. No, it is mainly involved in repair system. Third one is DNA polymerase 3, and this is the enzyme with high processivity, and this enzyme is the major enzyme which is involved in the replication. So option C is the correct answer. So thank you everyone for listening to our lecture. This is the last video of DNA replication series, and we will be back with transcription series. If you have any doubt regarding these slides or this course on DNA replication, please ask your queries in this email address, and you can text on this WhatsApp number too. So thank you everyone.